right, so yesterday we talked about correlations and we talked about sampling. Today we're going to be focusing on actual experiments. Now I know that you guys already read this in your book, and so some of this may be a slight redundancy, but at the same point in time, there are a few things in here that I want to make sure that we are clear on, especially moving forward with your guys' research projects. Okay, so first off, double blind. Okay, double blind procedure. The guy yesterday in the video kind of mentioned a double blind. Does anybody remember what a double blind actually is? What do you think? Experimental. So, with a double blind, you are going to have nobody knows, you don't have the experimenter, you don't have the participants know, neither one of them knows which one is control group, which one is the experimental group, okay? What's the point? Why do we do that? So there's no bias. What would be the potential bias of the participants? So they would act differently potentially if they knew going into it, oh yeah, I'm only getting a placebo. I'm not doing anything. Whereas if you're talking about actually receiving the treatment, all of a sudden they think, oh, look at that, I'm magically cured. Okay. On the part of the researcher, why do they do a double blind? What do you think? They can't accidentally what? How would this, I mean, the researcher, I mean, they wouldn't want to sway it. Why? How would they accidentally sway it? One way or the other? Okay, so how could they potentially be, I mean, usually researchers go in with the intention of, I will not be biased. I am going in with a clear head. I am not going to have preferential treatment for one group or the other. How would they unintentionally do that? Okay, so they could potentially be specifically nitpicking on the placebo group, trying to find faults with them. Or they could unintentionally, the way they are looking for, they may know, for example, if you're talking about drug trials are particularly tricky, okay, and they may be looking for, they know specific side effects that they're looking for, so they may be specifically looking for those things in that one particular group, but ignore the same symptoms in a control group, okay? So double blinds are purposely done that way. There's also a triple blind. Your book didn't talk about triple blind. Triple blind is going to be where not only the participants don't know, the researchers don't know, but you also have you have individual separate people that actually analyze the data, and they don't know what group is what. Okay, so somebody would look at the data and they would look at the actual information from it, and then they would not even know who's in the control and who is in the experimental group. Okay, so that would be an example of double blind there. All right, so different kinds of groups. First of all, experimental are going to be the ones that are receiving the treatment. Notice, remember yesterday when we watched that short video where he explained that whole caffeine experiment, and he had the control group that had decaf, and then he had two experimental groups, and he had one experimental group that had, like a regular cup of coffee, basically. And then he had another experimental group that actually had a higher dosage of caffeine. Okay, so he upped it, and what did he say? It was like 5 espresso shots or something like that. It was ridiculous. Okay, so you can have more than one experimental group, but they're going to be the ones that are receiving the treatment. And then, obviously, the control group doesn't receive the treatment. They have some sort of placebo. If it is a drug trial, okay, they will do that. Or they will, like for example, the caffeine thing, they had decaf. Or it'll be a case of, that the researcher will ask them maybe instead of giving them any kind of coffee at all, they'll just have them run the maze by themselves, okay, without drinking anything particular. You could give them water, okay? All right, so that was the example that they used is that the decaf, the regular coffee, and the super size. Okay, so variables. This is one part that I know that a lot of people get confused. Okay, independent variable. Okay, independent variable is going to be the factor that is changed. So if we're talking about the whole caffeine thing, okay, the independent variable would be the caffeine, the amount of caffeine. Because we're changing the amount of caffeine that each participant is actually consuming, okay, that's the independent variable. Whereas the dependent variable, okay, is going to be what we're looking at, what we're measuring as a result. Okay, the variable that changes as a result of the independent variable. So you can, remember this from the standpoint of that we are talking about a dependent variable. Dependent people, okay, they rely on others. They're kind of like a drag. They pull you down. Okay, so dependent variable is going to be a variable that changes as a result of the independent variable. So in the case of the, and this is where people get tripped up. Usually they can identify the independent variable, but not the dependent. So in our coffee scenario from yesterday, remember they were supposed to drink either decaf, a regular shot coffee, or like this super-sized coffee, and then they were supposed to run this maze. Okay? So, which one's the dependent variable? How fast, how long it takes them to run through the maze, right? Okay? So you've got to make sure that that's the part, because that's the part that people will get tripped up on as to what the dependent variable is. Okay? So make sure you pay attention to making sure that your dependent variable is actually measurable. Remember what he said yesterday is that you can't go and measure, for example, intelligence. Or you can't just say intelligence. You have to be specific. So if you want to measure intelligence, there could be IQ tests that you could measure. In this case, he talked about speed. Okay, he specifically gave them something to do and timed them. You must have a measurable way to measure the dependent variable. You can't just willy-nilly say that that's something that they're looking at. Okay, so the time was actually the dependent variable. Another example, if we're talking about the whole idea of yesterday I mentioned studying and grades. Okay, we wanted to do an experiment on studying and grades. Okay, so if I go and have some people go and study in a room that is full of noise. Okay, there's a TV on, there's music playing, there's a fan going, and I have people studying there for 2 hours every day. Okay? And then I have another room where it's complete and utter silence. What's the independent variable? The noise. What's the dependent variable? That's the tricky part. You said how well they studied. You can't measure that. Like their grade. Their grade would be a dependent variable. I would have to then go and give them a test. Okay? So if I give them something specific to study and then measured how they did on that test, then I can talk about the dependent variable. Okay, so moving on.